Welcome to Biology 101. In this video, we will be talking about cell communication. We will be covering the types of cell communication, the endocrine signaling process, and the types of receptors and signal transduction pathways which exist. There are four primary types of cell communication. The first type is autocrine signaling. Autocrine signaling occurs when a cell signals to itself. An example of this is with a cancer cell. A cancer cell signals to itself to duplicate into more cancer cells, and those cancer cells signal to themselves to continue to divide into more cancer cells. Juxtacrine signaling occurs when there is direct physical signaling from cell to cell. We can see this in the animal and plant cells, which have cell junctions that directly connect their cytoplasms with adjacent cells, allowing them to communicate directly. So as we see here, this would be our cell junction, and now these cells can send signaling molecules to each other directly. Paracrine signaling occurs when messenger molecules are diffused to send signals to nearby cells. A prominent example of this is with neurotransmitters. Neurotransmitters are messenger molecules which are diffused between nerve cells. Finally, endocrine signaling occurs when hormones send signals along long distances to target cells. Now in this video, we will be focusing primarily on the endocrine signaling process. So how does endocrine signaling occur? Endocrine signaling occurs in four main steps. The first step of which is signaling. Signaling occurs when the ligand, or the signaling molecule, is sent from the original cell, as we can see here. Next, reception occurs when the signal is received by a structure called a receptor, as we see here. The signal is received when the ligand binds to the receptor. Transduction occurs when the signal received by the receptor is amplified by secondary messenger molecules. So here we would see the signal being amplified or modified. Finally, response is the action or cellular activity that occurs as a result of the signal. So the signal is received, it's amplified, and that leads to cellular activity or response. Out of all these steps, reception is arguably going to be the most important step, because only cells with the necessary receptors are going to be able to produce a response in the first place. So for instance, in the model that we have below, if the receptors here did not exist, then the signal wouldn't be able to be transduced or produce a cellular response. Now that leads us nicely into the various different types of receptors. There are two main types of receptors. The first of which is the intracellular receptor, which is located on the inside of the cell, and the second of which is the membrane receptor, which is located on the cell surface or membrane, as we can see here and here. Now there are three different types of membrane receptors, which include ligand-gated ion channels, G-protein coupled receptors, and enzyme-linked receptors. So, let's take a closer look at these various types of receptors and see how they affect the cell communication pathway, starting with intracellular receptors. As we noted before, intracellular receptors are located within the cell. Consequently, this also means that the ligand must also be able to pass through the lipid bilayer of the cell membrane. Now, if you don't remember, the lipid bilayer looks like this. Now, the lipid bilayer has hydrophilic heads and hydrophobic tails. Now, you can see why it might be a problem if a hydrophilic or a polar molecule tries to pass through. It wouldn't be able to because the molecule will be repulsed by the hydrophobic tails. As a result, generally, the ligands of intracellular receptors are nonpolar and hydrophobic. Now, once the ligand is able to pass through the membrane, it also has to bind to the intracellular receptor itself, which typically has somewhat of a complex structure. This structure includes the transcription activating domain, which is responsible for activating DNA replication, the DNA binding domain, which binds to DNA, an inhibitor, which blocks the DNA binding domain from binding to DNA, and the ligand binding domain, which holds the inhibitor in place and binds to ligands. If a ligand binds to the ligand binding domain, it changes the protein's 3D structure, allowing the inhibitor to be removed, as we see here. Once the inhibitor is removed, this allows the DNA binding domain to be exposed and bind with DNA, which in turn allows for changes in genetic expression. And finally, these changes in genetic expression lead to cellular activity. Now, once again, you can see the 4 different steps of endocrine signaling here. The signaling occurs once the ligand is released from the original cell. Then reception occurs when the ligand binds to the intracellular receptor. Transduction occurs when the reception of the signal leads to changes in genetic expression, and the response occurs as a result of the cellular activity, which results from the changes within the replicated DNA. Now, you can see why if the receptor weren't there will be a major problem. If the receptor weren't there, then the ligand wouldn't have anything to bind to, and as a result, there wouldn't be any changes in genetic expression, leading to no changes in cellular activity. The next type of receptor that we're going to be talking about is the ligand-gated ion channel. Ligand-gated ion channels are membrane proteins, meaning they are located within the cell membrane, that facilitate the diffusion of ions through the use of what we call a concentration gradient. Now, these ion channels work when the ligand binds allosterically to the receptor, as we see here. This changes the shape of the channel protein, as we see the channel protein opening up, which allows for ions to pass through. Now, as we said before, the ions are able to pass through the channel through the use of a concentration gradient, meaning that there's more ions on the outside than the inside. As a result, ions generally want to go from areas of a higher concentration to areas of lower concentration, allowing the ions to pass through once the channel is opened. G-protein coupled receptors function by activating a membrane protein called the G-protein, as we see here. The G-protein coupled receptors work in a series of steps. At first, the G-protein is inactive as the ligand is not binded to the receptor. When the ligand binds to the receptor itself is activated, leading to a change in its 3D structure, allowing it to bind to the G-protein, as we see here. Now, once the G-protein binds to the receptor, it is also activated, causing it to exchange GDP for GTP, which is a higher energy molecule. And it also causes the protein's alpha subunit, as we see here, to split off from the rest of the molecule. Now, the alpha subunit binds to an inactive enzyme. The activated enzyme is able to activate secondary messenger molecules, as we see here. by hydrolyzing GDP into GTP. Finally, the secondary messenger molecules elicit a response and cellular activity. Once again, you can see the 4 steps of endocrine signaling: the signaling itself of the ligand, the reception, the transduction through secondary messenger molecules, and the response in cellular activity. Finally, enzyme-linked receptors are membrane receptors whose intracellular domain is associated with an enzyme. Sometimes this means that the intracellular domain is the enzyme itself, or interacts directly with the enzyme. Now, the most common example of an enzyme-linked receptor is what we call a tyrosine kinase receptor. Now, these receptors work when a ligand bonds two close by tyrosine kinase receptors. So we would have a ligand binding to this tyrosine receptor and a ligand binding to this one, causing them to dimerize or combine together. As a result of the dimerization of the tyrosine kinase receptors, phosphates are able to attach themselves to the tyrosine residues located in the intracellular domain of the receptor. This then allows the phosphorylated tyrosine molecules to transmit the signal to other messenger molecules, leading to a cellular response. So what did we learn in this video? First, we learned about the different types of cell communication, which include autocrine, juxtacrine, paracrine, and endocrine signaling. Then, we covered the endocrine signaling process, which occurs in 4 main steps, which include signaling, reception, transduction, and response. Finally, we talked about the types of receptors and transduction pathways, which include intracellular receptors, located on the inside of the cell membrane, and membrane receptors, which embed themselves within the membrane and include ligand-gated ion channels, G-protein coupled receptors, and enzyme-linked receptors.